Hello everyone, welcome back. Today I'm here with this new presentation on bacterial reproduction. In this presentation I will try to discuss about the mode of reproduction in bacteria, that is asexual reproduction and sexual reproduction. Bacteria reproduces asexually with the help of binary fission and by the formation of endospore, while true sexual reproduction is absent in case of bacteria, but it shows certain level of genetic recombination with the help of some methods like we have bacterial transformation, conjugation, and transduction. So these are the topics that I will try to cover in this presentation. Let's dive in. So bacterial reproduction, दो ways से bacteria reproduce करते हैं. One is asexual method, other is the sexual method. So asexually reproduce करता है with the help of binary fission. जब conditions favorable होती हैं तो bacteria reproduce करता है binary fission की help से, और जब conditions टफ होती हैं तो बैक्टीरिया बनाता है एंडोस्पोर. वाइल जो सेक्सुअल रिप्रोडक्शन है, ट्रू सेक्सुअल रिप्रोडक्शन इज नॉट प्रेजेंट इन बैक्टीरिया बट बिकॉज दे लैक सेक्स ऑर्गन, बट जेनेटिक रिकॉम्बिनेशन टेक्स प्लेस. वहाँ पे जेनेटिक रिकॉम्बिनेशन होता है, मीन्स जो जेनेटिक मटेरियल दैट गेट्स एक्सचेंज बिटवीन टू डिफरेंट बैक्टीरियल सेल्स, और ये होता है विद द हेल्प ऑफ ट्रांसफॉर्मेशन, कॉन्जुगेशन एंड ट्रांसडक्शन. सो फर्स्टली एसेक्सुअल रिप्रोडक्शन. सो दो मेथड्स हैं. वन इज दिस वन, दैट इज बाइनरी फिजन. जो एक पेरेंटल सेल है उसमें फर्स्टली जो जेनेटिक मटेरियल है वो रेप्लिकेट करता है एंड फॉर्म्स टू कॉपीज, देन इट इज फॉलोड बाय डिवीजन ऑफ साइटोप्लाज्म एंड फॉर्मेशन ऑफ दिस कंपार्टमेंट, जिससे दो सेल्स बन जाएंगे, दो डॉटर सेल्स. सो दिस मेथड इज नोन एज बाइनरी फिजन, और ये होता है तब जब कंडीशंस होती हैं फेवरेबल. while the second method is by the formation of this tough structure which is called endospore. It do not involve production and fusion of male and female gamete, so that's why this is called asexual method of reproduction, and this is uniparental process. इसमें single parent involve होता है, और दो methods हैं. First is binary fission, other is endospore formation. So the first way to reproduce asexually is binary fission. As the name indicates, bi means two और fission means breakdown. इसमें जो एक bacterial cell है वो डिवाइड करता है into two daughter cells. So first step is replication of DNA, then there is formation of this septum. बाद में ये complete septum बनता है और separation होती है DNA और cytoplasm की. Then finally दोनों cells separate हो जाते हैं. That results in the formation of two independent daughter cells. और ये process follow करते हैं bacteria under favorable conditions. Then normally जो bacterial cell है वो डबल हो जाते हैं नंबर में विदिन 9.8 मिनट्स, बट इन केस ऑफ ई कोलाई इसमें लगते हैं 20 मिनट्स. और इसी स्पीड से जब डिवाइड करते हैं बैक्टीरियल सेल्स, दे मे रीच अप टू दिस नंबर, दैट इज 4.7 into 10⁻21 इंडिविजुअल्स weighing 2000 tons in 24 hours, which is a humongous number. बट प्रैक्टिकली ये पॉसिबल नहीं होता है बिकॉज देयर आर नेचर चेक्स ऑफ फूड, स्पेस एंड कंडीशंस. तो बैक्टीरिया को फेवरेबल कंडीशंस नहीं मिलती हैं, कॉन्टीन्यूसली डिवाइड नहीं कर सकते हैं. सेकेंडली जो बैक्टीरिया दे आर कंसिडर्ड टू बी इमॉर्टल्स बिकॉज जो बैक्टीरियल बॉडी होती है वो डिवाइड करती है इनटू टू सेल और कुछ भी रिमेनिंग पार्ट नहीं रहता है. सो दैट्स व्हाई दे आर कंसिडर्ड एज इमॉर्टल. इन नेक्स्ट स्लाइड देयर इज वन वीडियो शोइंग the animated video showing the process of binary fission.
Most bacteria reproduce by a process called binary fission. During binary fission, the parental cell divides to form two daughter cells. In the first step of binary fission, the bacterium replicates or copies its chromosome and attaches each identical copy to a separate location on the cell membrane. Unlike eukaryotic cells, bacteria do not need to dissolve a nuclear membrane or assemble a mitotic spindle, making binary fission very quick and efficient. Second, the cell elongates. Since the chromosomes are attached to different locations on the cell membrane, they separate from one another. Third, the bacterium pinches off at the center and forms a septum, a separating wall between the two compartments of the cell. Finally, the cell splits into two new daughter cells. These daughter cells are fully mature bacteria that can grow and, if conditions are right, continue the binary fission process.
Second comes endospore formation. So these spores are thick-walled spores which are produced by certain gram-positive bacteria. Specifically जो bacilli bacteria होते हैं वो ये spore produce करते हैं, and these spores are produced under unfavorable conditions. दो तरह के स्पोर होते हैं. वन इज़ एंडोस्पोर जो कि बैक्टीरियल सेल के अंदर की तरफ बनता है. सेकेंड वन इज़ एक्सोस्पोर जो कि बर्ड की तरह आउटर साइड को बनता है. बोथ आर रेजिस्टेंट टू टफ कंडीशंस. तो कुछ ग्राम पॉजिटिव बेसिलाई बैक्टीरिया प्रोड्यूस करते हैं रेस्टिंग सेल्स जिन्हें एंडोस्पोर बोलते हैं अंडर अनफेवरेबल कंडीशंस, जब कंडीशंस फेवरेबल नहीं होती हैं. एंड दिस इज़ अ थिक वॉल्ड स्ट्रक्चर जिसमें बाहर कोट होता है. Then it is followed by various outer parts, like we have outer membranes, cortex, then we have the cell wall. Inner to this it contains this protoplasm and DNA. So these are highly resistant structures, बहुत ही resistant structure होते हैं, durable होते हैं. Dehydrated cells, they can very less amount of water which can survive under extreme heat, freezing temperature, exposure to many toxic chemicals and radiations. तो ये सारी tough conditions को tolerate कर सकते हैं. And the genera which produces these endospores are Bacillus and Clostridium. तो ये जो बैक्टीरिया होते हैं ये प्रोड्यूस करते हैं. And next comes structure of endospore. So it consists of this outer covering जिसे बोलते हैं exosporium. It is thin and delicate. Then it is followed by a thick cover which is known as spore coat. जो spore coat है, it is followed by the next covering that is called cortex. Then it contains the core cell wall. There is a wall which is designated with the help of this red color circle. Then it is followed by this cytoplasm or protoplasm, and it contains central genetic material that is nucleoid or DNA. और ये जो spores हैं ये produce होते हैं जब conditions होती हैं favorable. And this structure जिसके अंदर ये spore बनता है that is called spore mother cell, या इसे बोलते हैं स्पोरोगोनी. So this is a complex structure with multi-layered coverings. The outermost is exosporium और ये होती है thin और delicate. इसके नीचे होती है spore coat जो कि several layers thick होता है, that is made up of proteins, and it results in a formation of thick structure. और इसी की वजह से जो spore होते हैं they show resistance against extreme conditions. Inner to this is a cortex. Then cortex is very large and is composed of सब्सटेंस जिससे बैक्टीरियल सेल वॉल बनी होती है इस पेप्टाइडोग्लाइकेन. सो कॉर्टेक्स बना होता है पेप्टाइडोग्लाइकेन से, या इसी को बोलते हैं म्यूरिन. एंड द इनर मोस्ट लेयर इज स्पोर सेल वॉल. इसके अंदर होती है स्पोर की सेल वॉल. देन इट कंटेन्स सेंट्रल प्रोटोप्लास्ट दैट कंटेन्स डीएनए और न्यूक्लियोइड. और इसके अलावा इसमें प्रेजेंट होती है सेल ऑर्गेनेल दैट इज 70S राइबोसोम्स. सो दिस इज द स्ट्रक्चर ऑफ दिस एंडोस्पोर. नेक्स्ट कम्स जर्मिनेशन ऑफ दिस एंडोस्पोर. सो जो बैक्टीरियल स्पोर होता है ये जर्मिनेट करता है जैसे ही कंडीशंस फेवरेबल होती हैं. देयर आर सर्टेन स्टेप्स विच आर फॉलोड ड्यूरिंग दिस प्रोसेस. सो जैसी कंडीशंस फेवरेबल होती हैं तो जो बैक्टीरियल जो स्पोर होता है जर्मिनेट करता है और इसमें बहुत सारी मेटाबॉलिक रिएक्शंस और मेटाबॉलिक चेंजेस आते हैं जब ये जर्मिनेट करता है. फर्स्टली जो प्रोटोप्लास्ट होता है दैट एब्जॉर्ब वाटर, और इसमें जैसे ही पानी अंदर आता है इट स्वेल्स अप and becomes active. There is leakage of one chemical substance that is called dipicolinic acid. तो जो ये picolinic acid है ये लीच आउट हो जाता है. Then a germination tube is formed. Then ये जो germination tube है ये बनती है इसमें, and it will give rise to a bacterial cell. And this germination में भी जो bacterial cell बनेगा it may be terminal हो सकता है, it may be lateral. So each endospore develops into a single bacterium. तो एक एंडोस्पोर से एक ही बैक्टीरियम बनता है. So this is the process of asexual production which occurs in bacteria under unfavorable condition. Then second type of spores are exospores. Exospores, as the name indicates, they are found outside of the cell. और ये जैसे budding होती है वैसे ये spores बनते हैं. So these are again vegetative cells which forms the spore to the outside, just similar to the budding. Exospore do not contain this acid that is called dipicolinic acid. इसमें absent होता है ये acid. और जो member हैं कुछ genus like we have Methylocyanus and strains of photosynthetic bacterium, there is Rhodomicrobium. ये जो genus हैं ये produce करते हैं exospores. These are again resistant to desiccation, heat desiccation, and UV resistant. This formation is initiated by appearance of a bud-like enlargement and surrounding capsule at one end. तो इसमें बड की तरह appearance दिखती है, and that leads to formation of this spore which is known as exospore. The next comes the sexual reproduction, or rightly we can say genetic recombination, because true sexual reproduction is absent because bacteria इनमें कोई भी reproductive organs नहीं होते हैं, but it shows certain level of genetic recombination. So mainly ये genetic recombination show करता है with the help of transformation, conjugation and transduction. तो ये methods हैं इसकी genetic recombination के. So genetic recombination in which nucleotide sequences are exchanged between two similar or identical molecules of DNA. तो दो जो cells हैं उनके बीच में DNA का exchange होता है, so that is called genetic recombination. This is used in case of horizontal gene transfer to exchange genetic material between different strains and species of bacteria and viruses. So इसमें जो gene transfer का method, that is horizontal gene transfer. Here true sexual reproduction is absent, but genetic recombination होती है with the help of transformation, conjugation and transduction. So the first way of genetic recombination is transformation, जिसे commonly bacterial transformation भी कहा था. इसमें जो एक bacterial cell है वो DNA को, जो free DNA होता है उसके particles को release करता है, that becomes donor cell, और जो दूसरा cell है that receives the DNA and gets transformed, so that is called recipient cell. And this phenomenon was first discovered by Frederick Griffith in 1928. So transformation involved the uptake of free DNA molecule which is released from one bacterium, जिसे कहते हैं donor cell, and received by the another cell which is called recipient cell. And this phenomenon was first discovered by Frederick Griffith in 1928 while working on bacterium strain which causes pneumonia, that is Diplococcus pneumoniae. He used the two strains of bacteria, that is S strain which stands for smooth, क्योंकि इसकी जो colony की appearance थी वो थी smooth और shiny, and this strain was virulent means aggressive or disease producing. तो ये जो strain है ये pneumonia cause करता है चूहे में, इससे उसकी death हो जाती है. While this R strain which is इसमें जो colonies होती हैं they are rough in appearance and this strain is non-virulent, ये disease cause नहीं करता है. जब चूहे को इस strain को दिया जाता है, so जो चूहा है उसमें pneumonia के symptom develop नहीं होते हैं. Then he repeated his experiment, और इन्होंने फ्रेडरिक ग्रिफिथ ने जो S strain है इसको heat kill किया. Heat kill करने के बाद जब इन्होंने इसे माइस में इंजेक्ट किया तो माइस के को निमोनिया के सिम्टम्स अपीयर नहीं हुए and mice survived. Then after that he repeated again this experiment and injected mice with the combination of this heat kill S strain and living R strain. तो जब इन दोनों का combination दिया तो इन्होंने देखा जो माइस है उसके अंदर जो माउस है उसके अंदर symptoms appear हुए निमोनिया के, and he observed that there was some transforming factor, कोई सब्सटेंस था जो हीट किल एस स्ट्रेन से लिविंग आर स्ट्रेन में गया, एंड आफ्टर दैट दिस लिविंग आर स्ट्रेन वाज कन्वर्टेड इनटू लिविंग एस स्ट्रेन. और ये इन्होंने कंफर्म किया, ही टुक द ब्लड सैंपल फ्रॉम माइस और ही ऑब्जर्व दिस लिविंग एस स्ट्रेन बैक्टीरिया इन दिस ब्लड. सो ही कंक्लूडेड दैट and there was some transforming factor that entered from heat killed S strain to living R strain and converted this living R strain into living S strain. So that phenomenon of transformation of living R strain into living S strain is known as bacterial transformation. So for this he used two bacterial strain. One was the S strain which is smooth or virulent strain and having serotype S1, S2 and S3, and the R strain which is rough strain, and this is non-virulent, means ये disease cause नहीं करता है, with serotype R1, R2 and R3. For this study, for his experiment, he used S3 strain and this R2 strain of this bacterial for his experiment. So he selected S3 and R2 strains for this experiment. So we'll understand with the help of this image. Firstly, he injected type 2, that is this R2 strain of this bacteria that was non-virulent. जब इसको माइस में इंजेक्ट किया तो माइस survived, इसको निमोनिया नहीं हुआ. Then he injected mice with this S3 strain of Diplococcus pneumonia, जो कि निमोनिया कॉज करता है, जिससे जो चूहा है उसकी डेथ होगी. Then it is followed by his experiment that he injected this heat-killed S3 strain of Diplococcus pneumonia into the mice, and he found जो mice है उसके अंदर कोई भी pneumonia के symptom develop नहीं हुए, mice survived. Then finally he put a mixture of living R2 strain and heat killed S3 strains. जब इन दोनों का कॉम्बिनेशन इस चूहे को दिया गया, ही ऑब्जर्व दैट जो चूहा है उसमें निमोनिया के सिम्टम्स डेवलप हुए एंड माइस डाइड, एंड ही फाउंड in the blood of mice these S strain bacteria, S3 strains bacteria. So he concluded that there was some transforming factor that enters from heat killed S strain to living R strain and converted this living R strain into the living S strain. So this is called bacterial transformation. So one form of bacteria gets transformed into another form. We will understand this phenomenon with the help of one animated video. So in the next slide we will look for that.
In an attempt to discover a method for destroying the bacteria that cause pneumonia, Frederick Griffith conducted experiments on 2 strains of the bacterium Streptococcus pneumoniae. One of these strains caused the death of laboratory animals, while the second was more benign and usually harmless. Griffith assumed that the virulence and harmlessness of bacteria depended on whether or not they produced a shell. One of the strains, referred to as S, smooth, produced a smooth shell composed of sugars. The second strain, referred to as R, rough, did not produce such a shell. Griffith conducted a series of experiments on mice in order to find a vaccine. First, he injected mice with bacteria of the S strain. All the mice became ill and died. Then, by subjecting them to high temperatures, Griffith killed bacteria of the S strain and injected them into mice. As he expected, none of the infected mice developed pneumonia. When Griffith injected the mice with live bacteria of the R strain, they did not become ill. However, when he injected mice with dead bacteria of the S strain and live bacteria of the R strain, that is, the benign strain, the mice developed pneumonia and most of them died. The bodies of dead mice that were given both the S and R strains simultaneously contained living bacteria of the S strain, but no bacteria of the R strain. Griffith concluded that bacteria of the R strain had taken the genes responsible for the formation of cells from bacteria of the S strain, had become virulent, and had passed on the ability to produce shells to the next generations. This ability proved to be permanent and inheritable. The phenomenon of passing genes from one bacterium to another is referred to as transformation.
The Griffith's experiment was first experiment in search of genetic material. This experiment was continued by 3 scientists, Avery, McLeod and McCarthy. They confirmed that जो transforming factor था, that was a DNA fragment. What they have done, they took the heat-killed S strain bacteria, and in one set of experiment they added proteinases. Proteinases are the enzymes जो protein को degrade करते हैं, and they observed that when this heat-killed S strain bacteria was added in this mixture of in the culture medium which contains R strain cells, they observed that there was appearance of S strain bacteria in this set. In second set they added RNAs. RNAs जो enzymes होते हैं, they are used to break down RNA. So this culture was free from RNA. When they added this mixture into this other set of culture which contains living R cells, they again observed that there was appearance of living S strain cells. In final set they added DNAs. DNAs ऐसे enzymes होते हैं जो DNA को break down करते हैं. So this set doesn't contain DNA. So ये जो heat killed S strain का set है, इसमें DNA present नहीं था. And when this mixture was added in this culture medium containing R cells, they observed there was no appearance of S strain. तो इसमें जो S strain के bacteria वो appear नहीं हुए. From this experiment they concluded that जो DNA है, DNA of heat killed S strain, so this was responsible for conversion of this living R strain into living S strain. So it proved that addition of DNA from S3 strain to non-capsidated R2 strain transformed the R2 strain into capsidated S3 strain. when DNA is along with DNA of S3 strain is added, the culture R2 strain was not transformed. जब इस कल्चर में DNA इसको ऐड किया गया तो DNA इसमें डिग्रेड हो गया, then this culture was not able to transform these living R strain bacteria into living S strain. So that proved that DNA was the material that transformed R strain bacteria into the living S strain bacteria. The next comes mechanism of transformation. So the ability of a bacterial cell to take foreign DNA is known as competence. इसे competence कहते हैं. और ऐसे cells जो बाहर के foreign के DNA को अपने अंदर accept कर लेते हैं, these are known as competent cells. और ऐसे cells जो foreign DNA को accept नहीं करते हैं, these are known as non-competent cells. So this is the basic mechanism behind the mechanism of transformation. So ability of a cell to gets transformed depends upon competence, and competence is the ability of a recipient bacterium to take the foreign DNA and DNA from the outer environment. So competent cells are those cells which can be transformed. ऐसे cells जिनको transform किया जा सकता means they can take foreign DNA. As cells called the competent cells. We can make, there are some cells like we have E. coli cell that is not competent, but जब हम इस cell को treat करते हैं calcium chloride और heat shock देते हैं, so it can becomes competent cell, means it can accept the foreign DNA. So this is the basic mechanism of transformation. As we know that transformation, that the donor bacterium sends this DNA to the recipient bacterium, and this DNA gets integrated with this recipient DNA and transform it. This is called transformation. So there are two steps. The first step is there are certain DNA binding receptors on the bacteria. जो रेसिपिएंट बैक्टीरिया होता है उसके सेल के सरफेस पे लगे होते हैं DNA बाइंडिंग रिसेप्टर्स, और डबल स्ट्रैंडेड जो DNA वो यहाँ पे बाइंड करता है, एंड आफ्टर दैट इट एंटर इनसाइड द सेल, एंड आउट ऑफ टू स्ट्रैंड्स एक जो स्ट्रैंड है DNA का वो ब्रेक डाउन हो जाता है, एंड द सेकेंड स्ट्रैंड दैट कंबाइंस विद प्रोटीन जिन्हें बोलते हैं हम SSBs, दीज आर सिंगल स्ट्रैंड बाइंडिंग प्रोटीन. और ये जो सिंगल स्ट्रैंड बाइंडिंग प्रोटीन होती है दे हेल्प्स इन बाइंडिंग ऑफ दिस सिंगल स्ट्रैंडेड DNA टू द रेसिपिएंट cell's DNA. So this leads to formation of one structure which is known as heteroduplex. So here DNA binding receptors on the competent bacterium binds to double stranded DNA. As the DNA enters, जैसे DNA अंदर enter करता है तो इसमें से एक जो strand that gets degraded and the other strand is coated with protein जिन्हें बोलते हैं हम single strand DNA binding proteins. Then in second step, single strand of donor DNA is integrated into the chromosome of recipient cell. That results in the formation of structure which is known as heteroduplex DNA with different alleles in the two strands. So इसके जो structure बनता है is called heteroduplex. Sometimes the DNA gets degraded by DNAs, so that type of transformation is known as unsuccessful transformation. While there is second type of transformation, that is transformation with plasmid. So a donor cell will replicate this plasmid and send it to the recipient cell, so that is again a stable transformation. The second type of genetic recombination bacteria is conjugation. This phenomenon was observed by Liederberg and Totem. They observed that one bacterium, that is the donor cell, can transfer its genetic material to the recipient cell through a tube-like structure that is called sex pilus. इस ट्यूब के थ्रू एक बैक्टीरिया दूसरे बैक्टीरिया को अपना DNA सेंड कर सकता है. That is called conjugation. And as we know that the bacteria contains extra chromosomal DNA that is called plasmid. So ये जो plasmid है इसकी कॉपी बनाता है बैक्टीरिया और इसको सेंड करता है रेसिपिएंट सेल में. So that cell which contains this plasmid is known as F positive or cell, और जो initially this second cell is F negative, but after receiving this DNA it becomes F positive. So this phenomenon was discovered by Liederberg and Totem in 1946. In E. coli में ये प्रोसेस देखा गया था. Details were given by Woolman and Jacob. इसकी जो details हैं they were given by Woolman and Jacob in 1956. It is a process by which genetic material is transferred from one bacterial cell, जिसे हम donor कहते हैं जो DNA देगा, to the another cell that is called recipient cell, और इसे हम female cell भी कहते हैं, while the donor cell is known as male cell. Though a specialized intracellular connection is present, जो ये intracellular connection present होते हैं इसे बोलते हैं sex pilus और conjugation tube. इस conjugation tube के थ्रू DNA ट्रांसफर होता है एक बैक्टीरियल सेल से दूसरे बैक्टीरियल सेल के अंदर. Conjugating bacteria are of two mating types. दो तरह के होते हैं. वन इज मेल टाइप विच डोनेट्स देयर डीएनए, दीज आर कॉल्ड एफ पॉजिटिव सेल्स. ऐसे बैक्टीरिया जिनके पास ये एक्स्ट्रा न्यूक्लियर डीएनए प्रेजेंट होता है, मीन्स प्लाज्मिड प्रेजेंट है, जिन्हें कहते हैं एफ पॉजिटिव सेल्स. एंड सेकेंड टाइप विच आर नोन एज फीमेल टाइप्स, मीन्स जो रेसिपिएंट सेल्स हैं जो इस डीएनए को रिसीव करेंगे थ्रू दिस कॉन्जुगेशन ट्यूब, दीज आर द सेकेंड टाइप सेल, जिन्हें हम एफ नेगेटिव सेल्स बोलते हैं. द एफ स्टैंड्स फॉर fertility factor. Here F stands for fertility factor or sex factor. This F factor can exist in two states. One is where the extra nuclear DNA exists in this form, that is called plasmid. In some cases this plasmid gets integrated with the main DNA. कई बार जो ये plasmid है ये main जो nucleoid होता है उसके साथ ये integrate करता है. There forms a structure which is known as episome. So this is known as high frequency recombinant cell. इस तरह के सेल को कहा जाता है high frequency recombinant cell. So lies freely in this F factor, that is plasmid, that lies freely in the cytoplasm and is replicated independent of the bacterial chromosome. Means ये जो bacterial chromosome है इसके independently ये जो plasmid है ये replicate करता है. Sometimes this get integrated in which the plasmid gets integrated with the bacterial chromosome. जो प्लाज्मिड है ये इंटीग्रेट करता है बैक्टीरियल क्रोमोसोम के साथ. दैट रिजल्ट्स इन फॉर्मेशन ऑफ वन स्ट्रक्चर दैट इज कॉल्ड हाई फ्रीक्वेंसी रिकॉम्बिनेंट, और इसे कहते हैं हम एपिसोम. फर्स्टली आई विल डिस्कस अबाउट कॉन्जुगेशन बिटवीन एफ पॉजिटिव एंड एफ नेगेटिव सेल्स. मीन्स द सेल विच कंटेन प्लाज्मिड एंड द सेल विच लैक प्लाज्मिड. तो इनके बीच में जब कॉन्जुगेशन होती है, सो दिस एफ पॉजिटिव विल रेप्लिकेट इट्स प्लाज्मिड एंड सेंड इट टू द एफ नेगेटिव थ्रू इंटरसेलुलर कनेक्शन, जिसे बोलते हैं कॉन्जुगेशन ट्यूब या फिर सेक्स पाइलस. So two bacterial cells come close to each other. Here in case two bacterial cell comes close to each other and the pilus of the F positive cell attaches with the F negative cell. तो ये जो conjugation tube है ये attach करता है दोनों बैक्टीरिया को एक दूसरे के साथ. Then double stranded fertility factor is nicked. जो ये fertility factor है it is nicked and begin to replicate. इसकी replication होती है. Produce single stranded copy of F positive factor. ये बनाता है single stranded copy of F positive factor and a forming double stranded factor, जिससे कि एक इसकी exact copy बनती है. And after that one copy migrates through the tube into the cytoplasm. जो इसकी दो copies बनती है उसमें से एक copy migrate करती है in the cytoplasm of F negative cell, जिसके पास ये plasmid नहीं था. It lies freely in the cytoplasm of recipient cell. और ये लाई करता है freely in the cytoplasm of recipient cell and both the cell become F positive, और दोनों सेल F positive आते हैं, means both the cell contain fertility factor. Now both the cell becomes donor or F positive. So this type of genetic material transfer from one cell to another cell is known as conjugation. In next slide we have one animated video, so that will make it more clear.
Bacterial conjugation is a process of genetic transfer between bacterial cells that requires direct contact between the cells. Many, but not all, species of bacteria can conjugate. Conjugation can occur between cells of the same species or even between cells of two different species. A small DNA circle or plasmid called the F factor is required for conjugation. The F factor stands for fertility factor. Strains of bacteria containing the F factor are called F+. Those without it are called F-. An F+ cell, or donor, produces a structure called a pilus to connect with another recipient cell. To begin conjugation, the F factor is cut at a specific region called the origin of transfer by a protein assembly called the relaxosome, which associates with the strand to be transferred, or the tDNA strand. Accessory proteins of the relaxosome are released, but a portion of the relaxosome called the relaxase remains attached to the tDNA. This tDNA relaxase complex is recognized by a coupling factor and transferred to the exporter, a complex in the F+ cell that is contiguous with the pilus. The exporter pumps the tDNA relaxase complex into the recipient cell. Once the entire tDNA molecule is transferred to the recipient cell, relaxase joins the ends to make a circular DNA molecule. As the tDNA is transferred to the recipient cell, it is replicated to become double-stranded. In the donor cell, the F factor DNA was also replicated to become double-stranded. This actually occurred as the tDNA was being transferred to the recipient cell. In the end, both cells wind up with a complete double-stranded copy of the F factor. Their connection through the pilus is released, and each is now an F+ cell that can go on to conjugate with other cells.
Next, there is a conjugation between high-frequency recombinant that act as a donor cell and the recipient F-negative cell, the cell which lacks plasmid. So here, there is a nick formation in this HFR DNA. इसमें एक nick बनता है जिसके बाद ये जो DNA है ये replicate करता है, and the small portion of this DNA gets transferred to the recipient DNA, and this portion gets integrated with this recipient DNA और ये receive करता है कुछ genes from this DNA और इसकी जो अपनी homologous genes है वो वहाँ से replace हो जाती है. That results in formation of this genetic recombinant. So this is the second type of conjugation that occurs between HFR, that is high frequency recombinant, and the recipient F negative cell, which is the female cell. Here, two bacterial cell, that is this HFR cell, and the cell which lacks this plasmid, that is the female cell, that comes closer to each other, and they form a tube, that is conjugation tube between these two cells. Then this circular DNA of this HFR gets nicked and start replicating. Here इसमें replication होती है, and this replication starts near the conjugation tube. ये होती है conjugation tube के नज़दीक, and this newly synthesized DNA piece gets migrated to the recipient cell. In nature, this mating of two cells exists for a shorter duration. ये जो mating होती है just थोड़े से time के लिए होती है, and it gets interrupted. This results in migration of small portion of DNA, क्योंकि ये थोड़े टाइम के लिए यहाँ पे इनमें conjugation होती है. So इस वजह से एक छोटा सा ही DNA का piece migrate करता है एक cell से, the donor cell से, to the recipient cell. That results in formation of this genetic recombinant. So newly entered gene may replace the homologous genes of the recipient DNA. तो यहाँ पे जो genes थी वो replace हो सकती हैं इस recipient DNA की, and results in the formation of this genetic recombinant. So this is the second type of conjugation that occurs between high frequency recombinant and the normal cell which lacks this fertility factor. The next comes third type of genetic recombination in case of bacteria, that is called bacterial transduction. So transduction is simply the transfer of DNA from one bacterial cell to another bacterial cell with the help of bacteriophage. जब bacteriophage की help से एक बैक्टीरिया से DNA ट्रांसफर होता है दूसरे बैक्टीरिया में, so that transfer of DNA is known as transduction, and this phenomenon was observed by Joshua Lederberg and Norton Zinder in 1952. तो जोशुआ लीडरबर्ग और नॉर्टन जिंडर ने ये phenomenon observe किया था in Salmonella typhimurium. इस बैक्टीरिया में इन्होंने सबसे पहले इस phenomenon को observe किया. So this is simply transfer of DNA from one cell to another cell with the help of bacteriophage. तो एक bacteriophage की help से एक बैक्टीरिया से जो DNA है वो ट्रांसफर हो जाएगा दूसरे बैक्टीरिया में, that is called bacterial transduction. And all phages can transduce, but not all bacteria are transducible. Means सभी बैक्टीरिया bacteriophage DNA को कैरी कर सकते हैं बैक्टीरिया के, but ज़रूरी नहीं कि सभी जो बैक्टीरिया है वो इससे ट्रांसड्यूस है. Means they can accept this DNA. Next about bacteriophage, as we know that bacteriophages होते हैं ऐसे viruses जो बैक्टीरिया को इन्फेक्ट करते हैं. दो तरह के होते हैं. वन इज़ वायरुलेंट, ऐसे बैक्टीरियोफेज जो अपना DNA बैक्टीरिया के अंदर ट्रांसफ़र करते हैं, एंड दे फॉर्म मल्टीपल कॉपीज ऑफ बैक्टीरियोफेजेस इनसाइड द बैक्टीरिया, एंड फाइनली दिस बैक्टीरियल सेल बर्स्ट आउट और इससे बहुत सारे बैक्टीरियोफेजेस बाहर निकलते हैं. सो ये ऐसे बैक्टीरियोफेजेस होते हैं वायरुलेंट, मीन्स दे कैन प्रोड्यूस डिजीज़ और इवेंचुअली डेथ ऑफ द बैक्टीरियल सेल. ये फॉलो करते हैं लिटिक साइकिल. देन सेकेंड टाइप वी हैव नॉन वायलेंट और टेम्परेट बैक्टीरियोफेजेस. ये जो बैक्टीरियोफेज होते हैं इनका DNA इनकॉर्पोरेट करता है विद द बैक्टीरियल DNA, और ये इस बैक्टीरिया के डीएनए को ब्रेक डाउन नहीं करते हैं। जैसे बैक्टीरिया का सेल रेप्लिकेट करता है वैसे ही वायरस का डीएनए भी रेप्लिकेट करता है। So in this way they forms multiple copies of this DNA, bacteriophage DNA, but under certain condition, कुछ साइकिल के बाद जो bacteriophage का DNA it may follow the lytic cycle. It means ये बैक्टीरिया का DNA को ब्रेक डाउन करेगा छोटे छोटे पीसेस में, and some pieces of bacterial genome gets enter inside the bacteriophage and that get transferred to another bacterium. So इस बैक्टीरिया से जो ट्रांसफर होगा DNA to another bacterium with the help of bacteriophage, that is called transduction. So in case of temperate bacteriophage, phage DNA incorporates into bacterial DNA, जिसे हम बोलते हैं प्रोफेज, रेप्लिकेट करता है इसके अंदर। कुछ साइकिल के बाद ये फॉलो कर सकता है वायलेंट साइकिल एंड कॉज लिसिस ऑफ दिस बैक्टीरियल सेल। सो दिस अकर्स इन केस ऑफ लैम्ब्डा फेज। ट्रांसडक्शन दो तरह की होती है, वन इज जनरलाइज्ड ट्रांसडक्शन और नॉन स्पेशलाइज्ड ट्रांसडक्शन। इट मीन्स बैक्टीरिया के डीएनए का कोई भी पीस, any fragment of bacterial DNA can enter inside the bacteriophage, means bacteriophage के अंदर होगा and it can transfer one bacterium to another bacterium with the help of this bacteriophage. So any piece, so there is no specificity. Second is restricted or specialized transduction. Here a specific segment of DNA gets transferred from one cell to another cell with the help of bacteriophage. So firstly we have this generalized or non-specialized transduction, as the name indicate. इसमें कोई भी रैंडम डीएनए का जो बैक्टीरियल डीएनए का फ्रेगमेंट है वो ट्रांसफर होता है बैक्टीरियोफेज के थ्रू, एंड दिस टाइप ऑफ ट्रांसडक्शन इज नोन एज जनरलाइज्ड ट्रांसडक्शन. So if all the fragment of bacterial DNA have chance to enter the transducing phage, the process is called generalized transduction. It mediates the exchange of any bacterial gene. Virus DNA enters the host cell and hijack its machinery to synthesize its own component. जो ऐसी बैक्टीरियोफेज वायरस को इन्फेक्ट करता है, इसका डीएनए होस्ट की मशीनरी को हाईजैक करता है एंड फॉर्म्स मल्टीपल कॉपीज, इसके डीएनए को तोड़ के अपनी बहुत सारी कॉपीज बनाता है. इनमें से कुछ बैक्टीरियोफेज के अंदर जो बैक्टीरिया का डीएनए होता है वो चला जाता है एंड आफ्टर दैट गेट्स ट्रांसफर्ड टू द अनदर सेल. So this transfer of bacterial DNA from one bacteria to another bacteria is known as transduction. So viral enzyme hydrolyzes the host DNA into fragments. Sometimes any fragment of host DNA gets incorporated in the virus. जो DNA होता है बैक्टीरिया का वो इनकॉर्पोरेट होता है बैक्टीरियोफेज के अंदर. देन सच बैक्टीरियोफेज इन्फेक्ट्स न्यू सेल एंड देयर इज ट्रांसफर ऑफ दिस DNA, और ये जो DNA होता है डोनर DNA, इट गेट्स इंटीग्रेटेड विद द रेसिपिएंट सेल टू फॉर्म दिस रिकॉम्बिनेंट DNA. So can transfer this to the donor DNA into new bacteria. It does not replicate inside the recipient bacteria but undergo recombination with the recipient cell. So that results in formation of this recombinant cell. This is called generalized or non-specialized transduction. We will understand it with the help of an animated video in the next slide.
In generalized transduction, a segment of DNA is carried from one bacterial cell to another by a bacterial virus called a bacteriophage or phage. The phage attaches to the bacterial cell and injects its nucleic acid into the host cell. A phage enzyme is produced that breaks down the host DNA into smaller fragments. Phage DNA is replicated and phage coat proteins are produced. During formation of the mature phage particles, a few phage heads may surround fragments of bacterial DNA instead of phage DNA. The phage particle carrying the bacterial DNA infects another cell, transferring the bacterial DNA to the new cell. When the bacterial DNA is introduced into the new host cell, it can become integrated into the bacterial chromosome, thereby transferring genes to the recipient. This cell then multiplies and carries new genetic material.
Then we have the second type of transduction, that is called restricted or specialized transduction. As the name indicate, इसमें specific DNA bacteria का transfer होता है through bacteriophage. So transfer of few restricted genes from donor bacterial cell. कुछ एक specialized gene transfer होती हैं एक बैक्टीरिया से दूसरे bacterial cell में. So carried out by temperate bacteriophage. ये carry करते हैं temperate bacteriophage. As we know that here we can see जो bacteriophage है इसका DNA अंदर आता है and it incorporated with the bacterial DNA to form the structure that is called prophage. And after some time, under certain condition, it can follow the lytic cycle. जब इसमें कुछ stimulant like we have chemicals or UV radiations, तो ये जो DNA होता है it can follow this lytic cycle. It will form multiple copies of DNA. जो DNA है Then after that it will break down the bacterial DNA into small pieces. So virus incorporates genome within the host cell DNA at certain location and remain dormant and passes to generation. When exposed to certain stimulus like chemicals, UV radiation, it follows the tick cycle. तो यहाँ पे chances है कि कुछ जो DNA है बैक्टीरिया का ये इस वायरस के अंदर चला जाए और ये ट्रांसफर करता है कुछ selected सा part with another bacterial cell. So this is called restricted or specialized transduction because certain part of the bacterial specialized segment of DNA of the bacterial cell gets transferred to the bacteriophage. That is called specialized or restricted transduction. This can be understood with the help of one animated video in the next slide.
Specialized transduction involves the transfer of only a few specific genes from one bacterial cell to another by means of a phage. The lambda phage, which infects E. coli, is a well-studied example of a specialized transducing phage. When lambda phage infects E. coli, the phage DNA enters the cell and then integrates into a specific site on the host chromosome. When an E. coli culture carrying the lambda phage is induced, phage particles are produced. On rare occasions, a piece of bacterial DNA, for example the gal gene, near the specific site of insertion remains attached to the phage DNA and a piece of phage DNA is left behind. The phage that develop are defective because they do not carry the entire phage genome but can still infect other cells. The defective phage can attach to another bacterial cell and the DNA can be injected. Both phage and bacterial DNA now become integrated into the new host chromosome. Only bacterial genes located near the site of integration of the phage DNA can be transduced, hence the term specialized transduction.
Then finally comes significance of this transduction. So it helps in transfer of genetic material from one cell to another cell and alter genetic properties of the recipient bacterial cell, shows evolutionary relationship between prophage and the host bacterial cell, and it helps in studying gene linkage. So this was all about for us today's discussion about this bacterial reproduction. I hope you will get some idea from this presentation. If you have any questions, queries, and any suggestions, give it in comment section. Thanks for watching. Have a nice time.